I think we're ready to get started this evening. Good evening and welcome. My name is Peter Trappa. I'm Dean of the College of Science. And on behalf of the entire College of Science and our partners on this event here tonight, the Museum of Natural History, the Department of Biochemistry, and UHealth, I'm delighted to welcome all of you to this very special Frontiers of Science lecture. We have a fantastic talk on tap, of course, tonight from Dr. Venki Ramakrishnan, who's going to talk about my adventures in the ribosome. And in a moment, he's going to get a proper introduction. But I thought I'd begin with saying just a few words about the history of the Frontiers of Science lecture series. The Frontiers series, some of you know, some of you may not know, is the longest continuously running lecture series on campus. It was launched in the 1960s by physicist Pete Gibbs, who had the idea of bringing together world-class scientists and a general audience. Now, the title, Frontiers of Science, was a nod to a famous white paper written by Vannevar Bush. Vannevar Bush went on to become president of MIT. This white paper was entitled Science, the Endless Frontier, and it laid out a very clear rationale for the importance of basic scientific research and the importance of supporting it and funding it, and in fact, it became the basis for the creation of the National Science Foundation. Well, in any case, in the intervening years from the launch of the first Frontiers lecture, the series has seen a great number of very distinguished guests and lecturers. In fact, 24 of the lectures were delivered by Nobelists, and of course we're adding to that count tonight. But even so, I'm often reminded that Vannevar Bush's thesis about the importance of basic scientific research, it's as important today as it was all those years ago. If you look at the early dates of the Frontiers lectures, you see them conspicuously clustered in January and February. The transaction was pretty clear at that time. You get these luminaries to come out and give a lecture, and then they'd spend a couple days skiing at Alta with local guides. And in some ways, we're still trading on that formula. We're at this confluence of exceptional science and this very beautiful, spectacular place which we call home. And we're especially reminded of that in this beautiful house of science, the Natural History Museum. And it's appropriate that part of tonight's event is the opening of a new exhibit on the fourth floor about the ribosome and some of Dr. Ramakrishnan's most famous and impactful research. Let's see, before we get to the introduction of the speakers, I want to tell you just a little bit about how the events tonight will unfold. We'll have the lecture, of course, and then afterwards a brief Q&A. And then that exhibit on the fourth floor will remain open until the museum closes at 9:15 this evening. There are a great many people to thank for an event like this. I'll have to keep my remarks brief. I'd like to recognize Caitlin Goings on my staff, Jason Cryan, the director of the museum here, and his incredible team, and Wes Sundquist, and all the folks in biochemistry who made this possible. So without further ado, I think I'd like to turn the podium over to my friend and colleague, David Goldenberg. He's a professor in the School of Biological Sciences, and he's going to give our speaker a proper introduction.
Thank you, and thank you everyone for joining us tonight. So it's a great pleasure, honor, to introduce Dr. Venki Ramakrishnan as the Frontiers of Science lecturer. Those of you who have attended any scientific presentations know that that's how all introductions of speakers begin, but this one is really an extraordinary pleasure and honor because Venki is an extraordinary scientist and individual, and he also has a deep connection here to the University of Utah, where he was a faculty member in the biochemistry department for several years. And well before he was known as a Nobel laureate, he was known by colleagues as a thoughtful, insightful scientist and a warm colleague who was greatly valued here. So the second part of an introduction like this is usually a bit of a short biographical accounting of the speaker's training and experience. But tonight, this rather unusual lecture, Venki's going to, I think, do most of that for us. So I will not try to cover his biography, but instead just emphasize what I think is really outstanding about Venki and his scientific career, which is that early in his career, late 1970s, he identified a really crucial problem, the question of the structure of this remarkable machine that makes proteins. And he stuck with that problem for decades. But in the course of that, he also stood out by taking bold leaps when it became clear that something new had to be done to really make progress. And one of the first steps of that, which you'll probably say more about, was deciding that X-ray crystallography was the tool that he needed to adopt. And that had a variety of consequences, one of which is that he took a sabbatical at the Laboratory of Molecular Biology in Cambridge, England, the LMB, and shared a bench with a young postdoc named Wes Sundquist, who was later going to be hired in the biochemistry department and would eventually lure Venki here to Utah. So that had great consequences. The second bold step that he'll describe to you was to try to solve the structure of not just pieces of the ribosome, as he had been before, but to go for the whole thing. And that was a huge leap of faith and based on a deep understanding of what would make that possible, which was not obvious to many people at that point. So those are the things that I would highlight to watch out for in his probably very modest presentation. The third part of a standard introduction is a list of honors that the speaker has received. And this is the part where I had to actually make some notes, and it could go on for a very long time. So I'm just going to highlight a few of them. In 2003, Venki was made a Fellow of the Royal Society of London, the oldest scientific society, at least in the Western world, and is viewed, at least in Britain, as something more distinguished and exclusive than the US National Academy of Sciences. But the US National Academy caught on quickly, and in 2004, he was made a member of that very exclusive society. He also is a member of the Indian National Academy of Sciences, and in 2009, as you know from the information that's been spread, he won the Nobel Prize for his work together with Tom Steitz and Ada Yonath. In 2015, he was elected the president of the Royal Society, where he got to deal with a variety of interesting issues, including the Britain leaving the EU and the consequences of that for the British scientific community. There are a couple of other honors that he's received that he tends not to put on his CV and maybe even hide. In 2012, he was knighted by Queen Elizabeth, and then in 2022, oops, I've done damage already. I'm sorry. Oops, you're going to have to get restarted. I'm sorry, Venki. He was made a member of the British Order of Merit, which is an even more exclusive secret society that doesn't carry with it a title, but is even higher. So at the risk of him not speaking to me again, it's my pleasure to introduce Sir Venki Ramakrishnan.
So thank you very much for that kind introduction, David. And I want to say I'm really touched to be here. It's a real pleasure, and I want to thank all of the people who made this event possible. It started off with an idea by Ray and John to have a ribosome exhibition, and it required the participation of many people, including the Natural History Museum. And at the same time, David asked me if I would be willing to give the Frontier of Science lecture. So all of this came together. And I'm very touched to be here because, as you will see, a lot of the reason that I'm known is for work that began while I was here in Utah and where the first breakthroughs were actually made here. So I've got a lot to cover, so without further ado, I just wanted to talk about this slide, which has been up for a while, so you've all seen these people, and you're probably wondering what these sort of a poet and two novelists or writers and some composers have in common, besides being dead white European males. And the thing that they have in common is that they all died very young, and they died of infectious disease. And the COVID pandemic made us realize, that nature, we think we're sort of immune to nature, but nature doesn't care. And in fact, a bacterial pandemic, which would spread with no antibiotics available, would be very, very serious. It would make COVID look like a walk in the park. And this is from 2016. This is the cover of The Economist talking about the rise of antibiotic resistance. And so the question is, how do a lot of antibiotics work? And it turns out that many, in fact, almost half of the known sort of major classes of antibiotics work by blocking protein production in bacteria. And so that again leads to the question, why are these proteins so important? And how are they made? And you have to know how they're made to understand how they're blocked, okay? So during the course of this talk, I'm going to show you lots of molecules, and I'm going to show them to you in different ways. And just to give you an idea of how that is, if you want to show people, you can have a very realistic representation, like a photograph, okay? But let's say you want to capture a sort of essential caricature of the person, then you can get something like that. Still very recognizable, okay? And notice I'm being completely bipartisan here. Okay? And if you want to capture some symbolic essence of the person, then you've got Doonesbury's version on the right. You have a waffle or, what they call it, all hat and no cattle. Okay, that's the expression. Actually, Jim McCloskey would have appreciated this, so. Anyway, you can do the same with molecules. The molecules are made of atoms and organized in a specific arrangement. And this is, the all-atom model, with what's called a space-filling model, each atom occupying the correct volume. But if you have a very large molecule, it's just a mess of balls, okay? And this should be on, right? Okay. And so it often isn't possible to tell what's going on if you see this just big mess of balls. If you then make it more schematic so that you have lines between atoms which are connected by bonds, you get this sort of intermediate structure. And then you can have a very schematic structure so that the real molecule actually looks like this, but on the right, you have a very schematic structure which captures the essence of the molecule. And this molecule, of course, is DNA, and you can see right away it consists of 2 strands, and it consists of 4 types of building blocks, and these building blocks are sort of connected to each other from one strand to the one across it. So just bear that in mind when you see lots of different representations of molecules. So let's go back to how proteins are made. And the thing I want to say is we all have thousands of genes. This is actually a very old slide from 2001. It turns out we actually have only about in the low 20s, the 20,000s or so number of genes. And we don't have much more genes than a weed, Arabidopsis thaliana, or a worm, C. elegans, okay? They all have about 20,000 genes. So remember that anytime you feel like superior to lots of other life forms. And the question is, what do these genes do? Well, it turns out genes are units of information, and a lot of what genes do is contain information on how to make proteins. And when people talk about proteins, they think, oh, that's what our muscles are made of, or we need proteins in our diet. It turns out proteins do a variety of thousands of the functions in the cell, and I'm just going to show you a few of them. First, proteins are also long polymeric chains, and unlike DNA, which consists of 4 types of building blocks, it consists of 20 types of building blocks, actually 22, but 2 of them are rather unusual. And so you could represent them by letters of the alphabet. So often biologists will have a string of letters which represent the different amino acids, which have different chemical properties. Now, but what these chains do is when they're made, they'll fold up into a particular shape, and that shape is what gives it its function. Now, the shape it adopts depends strictly on the order of the amino acids that make up the protein. Now think a little bit about how miraculous this is. It would be like if I had little strips of paper, and on each strip I wrote down a different sentence. And based on what sentence I wrote, the strip of paper would fold up into its unique shape based on the sentence I wrote down. So it's that astonishing. But of course, it's all chemistry. And what it means is that some proteins look like these long filaments. This is collagen, and by weight, it's the most abundant protein in your cell. It makes up all your cartilage and connective tissue and so on. This is hemoglobin. It's the molecule that carries oxygen from your lungs through your blood to your tissues, and it's what gives blood its red color. And this is a molecule called rhodopsin, without which you wouldn't be able to see. It's a light sensor that sits in the membrane of cells in your eyes, in the retina of your eyes, and senses light, and then that gets transformed into a signal that is received by your brain. So just 3 different proteins, totally different shapes, totally different functions, but lots and lots of things like that. And every one of them is made by reading instructions present in a gene. So going back to DNA, here's the structure of DNA, double helix, and it has 4 types of building blocks. And the big discovery by Watson and Crick, apart from the nature of the double helix, was base pairing. And what that is that each of those bases has a unique partner. So an A will only pair with a T, and a C will only pair with a G, and vice versa. So the information on one strand actually dictates the information on the other strand. It completely specifies what the other strand should be like. And this shows you how you could get 2 from 1. And the strands separate. Each strand has the information to make the other strand. So you could get 2 copies of DNA where there was 1. And this solved the centuries-old mystery of how living things can duplicate and divide. Now, I told you proteins consist of 20 types of amino acids, and they're a totally different kind of polymer. So how would you go from DNA with this 4-letter alphabet to a protein, which is a sentence written in a 20-letter alphabet? And that involves a couple of steps. The first step is the relevant section of DNA, which codes for a particular gene, is copied. Now remember, the DNA in your chromosomes have thousands of genes. They're all sort of sitting next to each other, and the cell will only copy a particular gene when it needs it. Some of them are used all the time. Others are used in certain cells, but not others. For example, your skin cell makes very different proteins from your muscle cells or your red blood cells and so on. And this copy is a molecule very similar to DNA, except it's single-stranded, and its sugar is slightly different. Instead of deoxyribose, it's called ribose. It's got an extra oxygen atom, which gives it some unusual properties. The other difference is instead of a T base, it has a U base, but otherwise it's also a 4-letter alphabet. This course is called messenger RNA or mRNA, something relatively unknown until 2020. Okay, and then everybody started injecting mRNA into themselves, so we know what that is. And that mRNA contained the gene for the spike protein for the coronavirus, and your cell read that mRNA to make the protein, the spike protein, and presented it to your immune system. So how do you then go from, this is still a 4-letter alphabet, you have to get to a 20-letter alphabet. It turns out you have to read these bases 3 at a time, because if you read them 1 at a time, you could only code for 4 amino acids. You wouldn't be able to code for 20. Even if you read them 2 at a time, you could only have 4 times 4 or 16 combinations. So it turns out it reads the information 3 bases at a time, and because it's a code, it's called, the unit of code is called a codon. And it turns out there's a molecule, an adapter molecule, which is another kind of RNA called tRNA, or transfer RNA, because it transfers the amino acid to the growing protein chain. And at one end, it reads the 3 bases by base pairing, just like the base pairing between these strands of DNA. And at the other end, it brings along the amino acid. So for a particular codon, there'll be a particular amino acid, tRNA, that'll recognize it. Then for the next codon, there'll be a different tRNA. And then these amino acids get joined up, and you get a protein chain. And this was all figured out by molecular biologists. But while they were doing that, cell biologists were asking, where do newly made proteins end up in cells? And it turned out they end up in these blobby-looking particles, and you could actually purify these components and spread them out on an EM electron micrograph, and you could see these particles. And they all look very similar, and they looked like they were made of 2 halves, a large subunit and a small subunit. So we saw that all these particles, which seemed to be this place where newly made proteins were found, were these relatively large particles that consisted of 2 subunits. And these particles were isolated from a fraction called the microsome, and when they analyzed it, they found that they were about 2/3 RNA. So we have messenger RNA, we have transfer RNA, and now we have this particle's own RNA. So there are 3 different RNAs already that I've talked about, and I'll come later to why that's so, and lots of different proteins. So it's a highly complex particle, and because it had both RNA, which is ribonucleic acid, and proteins, and it was isolated from a fraction called the microsome, it was called the ribonucleoprotein particle of the microsomal fraction. Now, imagine if you had to say that all the time. And at some point at a meeting in the late 50s, Howard Dintzes, who spent much of his life at Johns Hopkins, said, look, why don't we just call it the ribosome? And that's the name that's stuck ever since. And you can see that, a lot of these proteins and RNA go to form each of the subunits, which come together to form the whole ribosome. Now, over perhaps 40 years, a worldwide community of ribosome biochemists worked out the fundamentals of what happens when the ribosome makes proteins. And the first thing one of the things they know is that the small subunit actually binds the genetic message, the mRNA molecule that contains the genetic instruction. And the large subunit is where the protein is actually made, the amino acids are joined up. So the first thing that happens is the ribosome has to find out where to start, but once it knows that, the first tRNA is positioned at the first codon, which is the first unit that it has to decode, and brings in the first amino acid. And the ribosome has these slots for these tRNA molecules. And so then the second tRNA comes into this slot, and if it's the right tRNA, that is, it matches this codon, so this tRNA has to match this codon, then it's accepted. But if it's not the right tRNA, it's ejected. And so tRNAs are coming in and out all the time, and it's only when the right tRNA comes in that it's accepted, and then the amino acids are joined together, and the ribosome moves along, and then third tRNA comes in, and it too is accepted only if it's the correct one, and then the second and third amino acids are joined. So you can see what's happening. The ribosome is moving along the mRNA and only incorporating the correct amino acids based on recognizing the code on the mRNA. And the joining of the amino acid occurs far away from where the code is read, because this is quite a large machine, as you'll see. So in the sort of 90s, this was sort of a more realistic picture which came out of electron microscopy of what the ribosome looked like. So instead of that cartoon, this is a slightly more realistic picture. The tRNAs are actually bent, they're L-shaped molecules. This is the mRNA, which snakes around a canyon in the small subunit. And this is the growing protein chain that comes out of the ribosome. And this is the tRNA that brings in the new amino acid. So I've told you a lot about the ribosome. And I want to now switch to how I got into the ribosome. So when I was 19, I graduated in physics from India, and I went off to do a PhD in physics in Ohio University, partly because it was one of the few places that would give me a fellowship with the kind of training I had, and also I think they didn't like the fact that my education was somewhat narrow, and I didn't take the GREs. But anyway, so I ended up in Athens, Ohio, and spent 5 years there, and this was the physics building. And while I was there, I think the only notable thing I did was to meet and marry my wife. And I can tell you what the problem was. So I had no trouble taking physics exams, and I passed all my exams. I passed my qualifying exams, which are like written exams in physics. But then when I started doing research, I just didn't have any sense of how to go about it. And so the thing is that problems in physics are very hard. And it's partly because physics is a much older and more mature discipline. You can think of modern physics as starting with Galileo, sort of 1500s and thereabouts. And, modern biology really started with genetics and in the late 19th century, and a lot of it really from the mid-20th century. And when I was doing my graduate work, there had been few really big breakthroughs in the last few decades. And by big, I mean like early 20th century physics, relativity, quantum mechanics, the structure of the atom, the nucleus, all of that stuff, discovery of really basic particles. And I thought if I stayed on as a physicist, I'd spend the rest of my life doing boring calculations that, would be publishable, but wouldn't actually add to real understanding. And, I'm not dissing physics, I'm dissing myself, okay? So I have to be clear about this. So what about biology? Well, at this time, I used to read Scientific American. I still read it from time to time. And every issue of Scientific American reported a major breakthrough in biology. For example, in the 70s is when people had figured out how to sequence DNA. They had figured out what structures of biological molecules look like, what our cell membranes look like, what the immune system, how the immune system worked, a lot of the fundamentals of neurobiology. All of this was happening. It was like a revolution going on, and it reminded me of early 20th century physics, except that it hasn't stopped. The revolution in biology is still going on. And I also knew that many physicists had made this transition, like Max Delbruck or Max Perutz, who founded the lab where I now work, and Francis Crick, who also worked in that lab. And so I thought of doing a postdoc, but then I thought, I don't actually know any biology at all. And if I go do a postdoc, I would end up knowing a very narrow slice of biology and wouldn't have really broad training. So I decided to go to graduate school all over again. And by this time I'd gotten married, and by the time I got my, about 2 weeks after I got my PhD, my son was born, and I had a 6-year-old stepdaughter. And my wife was very tolerant, so she didn't mind, my going back to graduate school with these 2 young kids. And so I rented a Ryder truck and, drove off from Ohio to San Diego. And this is where I spent 2 years at UC San Diego and learning biology. And any of you have read my book, there's some hilarious incidents there about what it's like to be a sort of ignorant physicist learning how to use a pipetteman. So by the second year, though, I'd done a bunch of rotations, I'd done a lot of lab work, and I sort of knew, and I actually took undergraduate courses because I couldn't understand any of the graduate courses, full of jargon. They were talking about lambda. To me, lambda was a wavelength. It was not, and they were talking about some virus that infects bacteria. So I actually took undergraduate courses like cell biology and genetics and biochemistry. And then in my second year, I thought, well, actually I know a lot of this stuff. And I came across another Scientific American article. And this one was written by 2 professors at Yale, one of whom had actually offered me a postdoc, but I turned him down saying, I don't know any biology, I'm not ready for a postdoc. And the other person who actually offered me a postdoc was Tom Stites, and I turned him down as well. But it's okay, he shared the Nobel Prize with me later. So after 2 years, I told my long-suffering wife, well, not that long. It's, we'd only been married a few years then. But anyway, I told her, I'd like to move again, this time to the other side of the country. And so there we are again, packed up and moved to New Haven, where I went to work for Peter Moore, who was one of Jim Watson's graduate students. And Watson's lab was really one of the pioneering labs for work on ribosomes. Actually, Ray Jesslin, I think, was in Jim Watson's lab as well. And there, we were using a physical technique to look at where all of the proteins were in the small subunit of the ribosome. Now, this was very satisfying at the time, and I'm very grateful to it because it's what introduced me to the ribosome, and I learned a lot of ribosome biochemistry. But even when it was finished, it didn't really tell us much about how the ribosome worked. Now, why is that? Well, to understand something, you have to see it at the right level of detail. So if you were a Martian hovering above the Earth, and you saw these little things moving along in straight lines in Salt Lake City, but rather curvy lines in Cambridge, England, you'd be sort of curious about it. And then if you got closer to it, you'd see this sort of, kind of machine-like thing, and it seemed to have wheels and has a steering wheel and something that, gets hot or something. And you still wouldn't understand it, but it's only when you see real detail that you would understand that there's actually an engine with pistons that fire, and ignite a fuel mixture, and then that power is transmitted through a crankshaft to the wheels, and then it makes the whole thing go. So you need to have the right level of detail to understand how something works. And it's the same thing with molecules. Now, the trouble with molecules is they're very small, and the question is, how would you visualize something that's small? Well, the typical way to look at small things is with a magnifying glass. So you have an object and you use a lens, and what the lens does is it collects scattered rays from the object and recombines them into a magnified image. And then you can look at the magnified image and see details. Okay, but there's a theorem in physics which says that you cannot see detail that's finer than half the wavelength of the light you're using. And it turns out the atoms in a molecule are about 1,000th or 1/10,000th the distance, the wavelength of light that you're using. So there is another kind of radiation which has the right kind of wavelength, and that's X-rays. X-rays and light waves are both photons, so they're actually the same kind of electromagnetic waves, but it's just that X-rays have very, very short wavelength. But there are no good lenses for X-rays, and also X-rays are damaging, so by the time you got enough signal from a molecule, you would fry the molecule, so you would destroy it before you'd even seen it. But there's another method, and that involves taking the molecule and forming crystals of it. And crystals are basically regular three-dimensional arrays. And then you take this crystal and you hit it with X-rays. And because the crystal is a regular array, these X-rays are scattered preferentially along certain directions. Now, this screen scatters light in all directions. No matter what seat you're sitting in, you'll be able to see the same information. But that's not true for crystals. The information is concentrated in certain directions because of a property called interference. But the point is, think of the lens. Even if you didn't have that lens, the information would be there. The lens is not creating the information, it's using it to recombine the information. So similarly, the scattered rays contain information about the nature or the structure of the object. You can measure the scattered spots, and then you don't have a lens, but what you can do is you can do in a computer what a lens actually does, which some of you will recognize as a Fourier transform. And what that computation does is, in the computer, you can generate a three-dimensional image of the object. And that's how crystallography works. I've glossed over a lot of detail, but that's sort of the basic idea behind crystallography. Now, by this time, I was a staff scientist, which is the equivalent of a faculty member at a national lab, at Brookhaven National Lab. And I was using this low-resolution technique, which I learned as a postdoc, and it wasn't going anywhere. And I thought, well, I really need to learn crystallography because that's the technique that's going to give me the level of detail needed to understand how biochemistry, really how molecules work. And so, my wife and I were, we liked Monty Python and, things like that. So we thought it'd be fun to spend a year in England. And also Cambridge, the MRC lab of molecular biology is where this technique had actually been developed for proteins. So I wrote to Aaron Klug, who was very famous, and he was a Nobel laureate, and he went on to become president of the Royal Society. And I thought, what are the chances that he's going to, want me as a sabbatical visitor? But fortunately for me, I'd crystallized a protein that was interesting to him. And so that was sort of the hook, and so he sponsored me for a Guggenheim Fellowship, and I spent a year there, and I learned crystallography. And then I should say, when I came back, I wasn't happy with the way DOE was really downplaying basic science at the national labs and treating them more as user facilities. And as you know, I met Wes Sundquist. I shared a lab bench with him when I was on sabbatical, and he was a postdoc. I didn't do any lab work, though. The only thing I did was I poured a gel, and over the next few months, Wes watched this gel dry out and eventually crack, okay? And so that was the extent. But I had actually brought 2 data sets with me, and I'd learned how to solve structures during that. And as a result of this connection with Wes, and I already knew about Ray and John's work on ribosomes, he invited me to Utah, and one thing led to another, and then I ended up joining the biochemistry faculty. Now, because of my sabbatical, I had an idea that you might be able to tackle very, very large structures like the ribosome using crystallography. At the time, it wasn't clear that you could solve something that large. Now, initially, it wasn't even clear that ribosomes were identical. Maybe they were different for different genes, for different mRNAs, et cetera. And now that idea is actually returning. There's some people believe that there's specialized ribosomes and so on. But the first sign that ribosomes were identical were these two-dimensional crystalline arrays that people saw in tissues, first initially in chick embryos, and then in these kind of lizard, tissues. And so that was two-dimensional. But the first real proper three-dimensional crystals was obtained by Ada Jonat, who was working with Heinz Gunther Wittmann in Berlin. And in 1980, they produced the first real crystals of the large subunit. This is the part which actually makes the protein from a bacterium. And then a few years later, a group headed by Marina Garber was able to crystallize the other subunit, the small subunit, as well as the entire ribosome. So by the mid-80s, there were crystals of the large subunit, the small subunit, as well as the entire ribosome. Now, these crystals didn't have good enough quality to give you an atomic resolution structure, which means the molecules weren't precisely aligned in the crystal. So in collecting the data, they would be blurred out, and so you'd get a fuzzy picture. But that would have been possible. Now, I want to just point out this guy, Marat Yusupov, went on to play a big role as well. So when I arrived in Utah, I had this idea, but this was the state of play. This is sort of what the ribosome looked like in 1995 when I came here. And 15 years after the first crystals, there'd been no progress, not even a low resolution, a fuzzy image of the ribosome. And in the meantime, Ada Jonat had gone on to get really good quality crystals of the large subunit. But many years had gone by and there were no structure of that either, no progress on that. And people were wondering if the ribosome is really too big for crystallography. Now, in the mid-90s, as a result of a couple of talks at ribosome meetings from Ada's group, a number of us felt that, well, maybe the field could use some new participants and some competition and new ideas. And it was sort of like the Mad Mad, I don't know if you've seen a Mad Mad World, where this guy talks about the treasure, and everybody pretends not to believe it, but they all independently go off secretly, in search of this treasure. So after one of these meetings, there were about like 3 ribosome groups, none of us told any of the others, but secretly we all went back to our labs and said, we're going to go for it, and that's sort of what happened. But the Yale group decided that they would work with the large subunit that Ada had already discovered and developed. And I thought, that's great. They can duke it out. And there were no good crystals of the small subunit or the whole ribosome. But I thought the whole ribosome was really too big. It's like, 2.5 megadaltons. In humans, it's 500,000 atoms. And so I thought that was too big. And I thought maybe we should focus on the small subunit, which is still very large, and it's 150 kilodaltons and so on. So I thought, well, let's go for 150,000 atoms. So I thought, let's go for that. So there I was in Utah, and I say this is in my Bin Laden days, so one thing I have to say is I'm very touched that the 2 graduate students who had the courage to start this work are both here. They're both now very distinguished in their own fields. This is Bill Clemens and this is John McCutcheon. And fortunately, at the time, they didn't know a whole lot about crystallography or really that much about the ribosome. And so they thought it was a great idea for a thesis project. Okay, and so sometimes it's good not to know too much, because then you think of 50 reasons why it won't work. Now this is Brian Wimberly, who later went on to play an absolutely crucial role in the analysis of and solving of the structure. But initially he was a little wary, but once things started, things we started making breakthroughs, he, luckily for us, joined the team. And so that was, and this is Joanna May, who was a technician, and Bob was working on Bob Dutnell was working on an unrelated project. And I want to point out, I'm the only guy wearing gloves when this picture was taken, okay? And so it was the days when I actually used to do some lab work. Now, we got started, but as soon as I got started, I got nervous. And the reason I got nervous is I knew that Ada Yonat had had crystals for 15 years. She'd had a group of 20 or 30 people. And I thought, this could, I don't even know how long it's going to take to get good crystals or any crystals. And, and then even if I have crystals, I don't know how long it's going to take to solve. I have some ideas, but, and then I imagined submitting a grant to the NIH saying, I don't have any crystals, but I have ideas for how to get crystals. And if I get crystals, I have some ideas for how I think I might solve them. And I could just imagine the study section just laughing, and throwing my grant in, into pile B. And so I thought, well, but at the same time, I thought, if I don't do this, other people will do it, and I'll watch it done, and it'll just be, it's a once-in-a-lifetime opportunity. So I'd done my sabbatical at the LMB, and so why is that institution so successful? So one of the things is that it has very stable funding. It's not dependent on 3 to 5 year grants. Actually, it's institutionally supported, and we're reviewed every 5 years, but it's not reviewed purely by anonymous review committees or externally. They actually come and visit you. You have a chance to argue about your progress and so on. So if you haven't made the big breakthrough, but they feel you're making progress, they'll keep funding you. And it's also an institution that had a long tradition of tackling important problems. So many of the projects there have taken 20 years or so to mature. People like Fred Sanger, who had about 40 papers, an H-index of 18, if you look by metrics alone, he might not get tenure at most places, okay? But he had 2 Nobel Prizes, and his top paper had 65,000 citations. So it doesn't do that. And of course, there was a big draw, in Cambridge, of the salary, the food, the weather, and the mountains. I have to tell you, some of them are not as bad as you might think, okay? But a couple of them, yes, definitely. So while I'd sort of, approached the MRC and basically decided that's where I was going to go, I decided really to stop almost everything in the lab and focus on the ribosome. And, that was, as I said, started by Bill and John. And very quickly, we got crystals that diffracted really well. And this was by paying careful attention to making sure the ribosomes were pure. Now, around this time, I attended a meeting. And at the meeting, I found out that Ada Yonat had given up on the large subunit, which was her big baby up to that point, and had obtained better crystals of the small subunit. And so now this was the exact problem that we were going to work on. So I was going to go be head-to-head with Ada Yonat instead of having my own little niche and having other people fight it out. And this was a Yale group working on the large subunit. And in the meantime, Harry Noller, who's a very famous ribosome biochemist, had hired Marat Yusupov and his wife. These were part of the team that had crystallized the entire ribosome in Russia. So they had brought to Harry's lab the know-how for how to crystallize the whole ribosome. Now, Harry wasn't a crystallographer, but he had the good sense to hire a really brilliant crystallographer, Jamie Kate, who, by the way, had initially, with another RNA molecule, worked out the strategy, the exact strategy that I had been considering for the whole ribosome. So when I saw that Jamie Kate had, joined Harry's lab, that was like, I was freaking out because I knew Jamie would have exactly the right approach to solving it. So suddenly, here we are in the middle of a 4-way race. So the one thing you shouldn't do in the middle of a race is move, because, you're going to lose time, you have to set up a new lab, it's going to be a year, and you should never move. And even more, you shouldn't move to a different continent, because then you have all sorts of other problems. So what did I do? Dana said, we can't match your salary because you're getting about half the salary you're making here, but I'll give you a raise anyway to make it more painful. So we moved to England in the middle of this race, but we had collected data even before I left Utah, and that led to our initial breakthrough, which is, we were able to position a number of proteins and interpret about a third of the molecule, but this was still at low resolution. It wasn't at atomic resolution. And then Bill and Brian Wimberly joined me in Cambridge, and then I also had another postdoc and another graduate student. And so I reassembled a team there, and it was a frantic year, because by now, all the groups had made these initial breakthroughs, so it was a flat-out race. And we had to use a number of these very powerful X-ray sources called synchrotrons, which are high-energy accelerators, so they accelerate electrons at very close to the speed of light. And so I often joke that you could join a ribosome lab and see the world, because you could go to near New York or Chicago or near the Alps in southeastern France, or to this place in northwest England. And but actually, you only see the world's synchrotrons, because when you're given 2 days, you're really working round the clock. And what they're seeing is this thing, this little, what's called a hutch. It's really a little room, but it's isolated, and you have to get out of there when you turn on the X-rays. And so what happens is this little pipe here is what brings the X-rays from that large ring of particle accelerators, which generate the powerful beam. And this is where your crystal sits, and this is like an electronic detector which detects the scattered X-rays. Now, when you get the experiment works, what you don't get is a structure. You get an image of the molecule. It's essentially a three-dimensional image, and in order to interpret it in molecular terms, it's a bit like solving a complicated three-dimensional jigsaw puzzle. So here's the image of the molecule, and what you do is, like with a jigsaw puzzle, you look for areas that you can recognize. So here's an area that people in the field would recognize. These look like two strands, and they're these regular bumps, and they seem to be somehow connected. And so if you know anything about RNA, this is a double helical portion of RNA. RNA can also form double helices like DNA. And so here you have sugar and phosphate and these bases pairing across with each other. So suddenly you've taken a piece of the image and interpreted it in chemical terms. And that's what you do. That's only a little piece of the image, and you end up doing that for the whole thing. And you end up, basically, it's like solving the entire jigsaw puzzle until there's no pieces left to solve. And that's then when you declare that you're done with it. And using just this method, the Yale group solved the large subunit at about the same time that we had solved the small subunit. And it turns out that the key parts, the parts where the amino acids are joined to make the protein chain, or the parts where the genetic code is recognized, they're all surrounded by just the RNA part of the ribosome. And this explains a very old dilemma. So when the ribosome was discovered, it was known to be made of both RNA and proteins, and it's the protein-making factory. So people thought, if it's made of proteins, how did the ribosome even get started? Because you had to have proteins to begin with, and it's the thing that makes proteins. So Francis Crick first had the idea that maybe early ribosomes consisted only of RNA. And then as the ribosome became more sophisticated, some of the proteins it made stuck to itself, and it sort of evolved to be this more complex creature. And a lot of biochemistry, notably by Harry Noller, pointed in that direction. But this was really the first direct proof that the active sites were essentially all RNA. Now, I started off with antibiotics, and the point is that once we had the crystal structure, we could see directly how antibiotics bound to the ribosome. And this helped us understand not only how antibiotics work, but also how the ribosome worked, because they blocked ribosome function. And so we could learn quite a lot about the ribosome from these antibiotic structures. And then it took us a few years to get a high-resolution structure of the entire ribosome with tRNAs bound. There had been a lower resolution structure from Harry's lab with, Marat Yusupov and Jamie Kate and others. But this was really the first structure with the mRNA and tRNA bound. Jamie Kate had a structure of the empty ribosome at high resolution just before this structure. And then over the next 10 or 15 years, we ended up kind of taking snapshots. The ribosome is like a machine. And if you want to understand how the machine works, you have to take a snapshot of the ribosome as it's doing different things so that you can follow how the ribosome moves and how tRNAs come in and how the tRNAs move through the ribosome and how it terminates and so on. And this took us a long time, and each structure, obviously we did the easiest ones first, and then progressively it kept getting harder. And then, in 2009, I got this phone call, which I didn't believe, partly because I'd had a fight with a guy in Sweden, a ribosome guy at a meeting in Sweden. And about 6 months later, it turned out he was on the Nobel Chemistry Committee. And so I thought, well, I'm never going to get this Nobel Prize. And I turned down all invitations to speak in Sweden because I thought, what's the point? They're not going to give it to me. So then when I got this call, I thought that I had two friends, a guy named Rick Wobe and another guy you might know named Chris Hill. And I thought these guys had engineered some Swedish friend of theirs to call me, So I told the guy, you have a very good Swedish accent. And, and so there was this huge burst of laughter. And then I realized I was on a speakerphone at the other end, And so, but then I actually said, well, if this is true, I want to speak to this guy, Måns Ehrenberg. And he came on the line. And so that's when I realized it was real. Now, given my background, I went to my local, town university in India, and I went to, not a major, Ohio University is not a major research university by any means. So it is really surprising how my life worked out. And anyway, the next day, Nobel Prizes are given for chemistry, physics, physiology, and medicine, etc. But clearly, the next day, I realized that the newspapers had realized that the important prize was the ribosome, because, there's Tom Stites and me featured on the front page of the newspaper. Of course, it had nothing to do with this Crown Princess Victoria sitting between us. Now, lots have changed, so, today you would not do this. It would be like just today, you would not play an 8-track cassette. Many of you don't even know what that means. And today, people use a different method called electron microscopy, which was discovered by this German scientist named Ernst Ruska in 1933, right when the Nazis had taken over Germany and just before World War II broke out. And by the time World War II was finished, lots of people were using electron microscopy, and they'd forgotten that Ernst Ruska had actually invented it. And so it took him 53 years to get the Nobel Prize, and you have to be alive to get it, and he died 2 years after he got the Nobel Prize. So the moral of the story is, if you've done something important, make sure you eat healthy and exercise, and so you can live long enough. And what this does is it allows us to look at ribosomes which are not abundant, which are heterogeneous, and so we can solve all kinds of ribosomal complexes that we could never dream of using crystallography. And in fact, a bare bones ribosome structure, the kind of thing that led to a Nobel Prize, today would be like a rotation project for a graduate student. So that's how fast the science has moved. So I want to close with a couple of things. One is, why did my life even work out? I think One is that I kept my options open. So if physics didn't work out, I was willing to go backwards and, switch to biology, or if I found I was at a dead-end technique, I would go off on sabbatical to learn a new technique. And I would move depending on where I felt the opportunities were best, for both my work and also my personality. My colleagues here will know that I'm kind of a nervous type. Anyway, the other thing is not to be afraid to ask for help and, to talk to people. And one thing is, everybody has imposter syndrome, and outsiders, if you've not been to major research institutions, you're something of an outsider. It can be geographic, or if you're a minority, or if you're, a woman in science, you may feel more of an outsider because you don't sort of fit what's called the norm. And so you may have more of an imposter syndrome, but you have to remember everybody has it unless they're sociopaths. So that's something to keep in mind. And finally, Max Pruitt said, science is not about talent. You need a lot of things. He called it the 4 Gs for the German words. So it's Geld, or money, and Geschick, or skill, which is also training and expertise, and Geduld, or patience, so not giving up at the first sign of difficulty. And everybody, once they're successful, they forget the Glück part, which is luck. All of us have had luck at key points in our career, meeting the right person, having somebody tell you an idea, or point you in the right direction. So we shouldn't forget that. So the real star is actually the ribosome, and thanks to Janet Iwasa, I want to just show you a movie of how the ribosome works. So we're starting off with the small subunit, which is going to bind mRNA, and that green is the start codon, the first codon, and it requires a number of proteins and that first tRNA, and then the large subunit binds, and then tRNAs are delivered to the ribosome, and then the tRNAs move, and the amino acids are added to a growing polypeptide chain, and so this is in slow motion, and you can see how Janet has speeded it up to roughly real time in bacteria, and that's what it would look like in real time. It's an amazing blur just to think that this 500,000 atom molecule is moving at that speed to make proteins, and this keeps on going and adding amino acids until it reaches a stop signal, and then a particular protein then cleaves off the newly made protein, and it goes off and folds and goes and does its thing, and then the ribosome is dissociated by other factors, and the whole process can start over again. And during the time you've been listening to me, the thousands of ribosomes in each of your cells have been churning out tens of thousands of proteins as we speak. And so, thank you very much.
Well, thank you, Venki, for a wonderful lecture. As I mentioned at the outset, we have time for a few short questions, and there are folks circulating with microphones. I'm told that in the School of Biological Sciences, preferential treatment is given for the first question if they come from undergraduates or graduate students or postdocs. So, although these lights are so bright, it's hard for me to identify anybody. So anybody has a question can raise a hand. There's one right back there, and we can probably quickly get a microphone there.
And could you
Yes,
could you speak a little slowly, because
Is it on?
I have terrible hearing and I have hearing aids, so
that's all right. Can you hear me?
yes.
Okay. I was wondering if there's any evidence of the ribosome changing, or can we see if it can change in an organism's lifetime?
I don't think it changes, obviously, as you saw in the movie. It changes its conformation and so on. I don't think it changes during the lifetime of the cell. But there is a debate going on in the field about whether there are things called specialized ribosomes, where some cells have particular types of ribosomes to translate particular genes. But that's still very highly debated.
Thank you.
What would you think it will be like the future, in your perspective, like the future of new knowledge about ribosome and translation and all these machineries?
I think where the field is going now is to understand how ribosomes are being regulated. Because it turns out we want to regulate gene expression. You don't want proteins to be made when they're not needed, and you also want proteins to be turned over when they're no longer required. And so the cell has very, very elaborate mechanisms to control when translation occurs and how much translation occurs. So that regulation of translation is probably one of the big key questions. A second key question is, how are ribosomes assembled? And how is this incredibly huge machine assembled? And it turns out in humans, there are over 100 proteins whose only job is to help. They're basically assembly workforces which help to assemble ribosomes into a functional form. So that's another big area.
Does antibiotic resistance have a ribosomal basis?
Sure. As I showed you, many of these antibiotics work by directly binding to the functional sites of ribosomes and preventing it. So some antibiotics will prevent the tRNA that brings the new amino acid from binding. Other antibiotics will prevent the amino acids from being joined into a peptide bond. So that depending on where they bind, they'll stop some key function of the ribosome.
But the resistance, the antibiotic resistance, does that have...
Yes, that's true too. You can get resistance by mutations in the ribosome, so the antibiotics won't bind in that pocket because the pocket will have changed its shape as a result of the mutation. But actually, many ribosomal mutations come about not because of those mutations, but because they're modified. The ribosomal RNA gets modified, and that causes resistance because the modification attaches extra groups and changes the shape of the binding pocket. And a third mechanism, which is also very common, is the cell turns on pumps, which pump out the antibiotic. So the antibiotics enter the cell, but the cell pumps it out before they can do any harm. The bacteria pump it out. So there are many mechanisms that bacteria have to evolve resistance. And the one thing you can be sure is whatever antibiotic you develop, eventually resistance will develop.
Hi, first of all, thank you very much. That was a beautiful presentation, and thank you for your career. It's a spectacular molecule. My question is this. Almost all of the other mechanisms of life have been replaced by protein, almost entirely. The polymerases, helicases, all the other stuff.
Yeah.
And yet the ribosome persists.
Yes.
What does it say about early life? You alluded to this. What does it say about early life? What does it say about the mechanism that it has survived across billions of years?
Yeah, so I have to tell you, so mitochondria, for example, human mitochondria, instead of being 2/3 RNA, are actually 1/3 RNA and 2/3 protein. So their RNA component has shrunk to really the essential core part of the RNA. So once I was at a meeting with Harry Noller, who's very much an RNA world person, And when you see the structure of the ribosome, you see all these proteins with snake-like extensions into the ribosomal RNA. And I kind of teased him. I said, you know, in a couple of billion years, these things are going to take over, and you're going to have a protein machine. And he said, yes, it's going to be like these cyborgs. You know, but anyway, one reason could be that, you know, how the ribosome evolved is still very, very unclear. But people like Harry, who've thought about it, and others, think that maybe the large subunit evolved first. It was just an RNA machine, and it just added amino acids in some random way, so that there was no genetic code and there was no tRNA and so on. There was just maybe short ends of tRNA that brought in the amino acids. And so you had this machine that linked up peptides, you know, random peptides. And gradually it evolved. And the tRNAs and the small subunit probably co-evolved with the genetic code and the gene. And so you had this co-evolution that resulted in this machine, and maybe that makes it a little harder to just gradually replace it, because it's multi-component machinery. It could also be that RNA has some properties, for example, for binding RNA and for tRNA, base pairing, all of that stuff. So it's particularly suited for this kind of recognition and chemistry, and it would be hard to replicate in a protein.
Maybe one more question from gentlemen.
I apologize, I don't have a background in biology, so I can't ask a biology question, but I do have a background in mathematical modeling and the creative thinking process regarding mathematical modeling. So I'd like to ask you a question regarding that. When Thomas Kuhn wrote Structure of Scientific Revolution, he talked about paradigms and paradigm shifting. And when Edward de Bono wrote about the creative thinking process, he talked about outsiders bringing new information to problem solving. Could you give a little more insight in terms of whether or not your background in physics really was influential in terms of how you framed your problem solving processes?
Yeah, I think it probably helped indirectly. I should say, you know, for example, Tom Stites was not trained as a theoretical physicist, and you know, certainly Harry Noller was trained as a really classical biochemist, and their labs were also doing ribosome structure. So I think the physics background helped me in one or two key things. One is I wasn't afraid to go off and learn new physical techniques like crystallography. Even relatively late in my career, I was 40 years old when I went on sabbatical. And so I think my physics background helped me there. What my physics background really helped me with was give me a sense of signal to noise. And, you know, it was a kind of a big leap, but I trusted my calculations. I knew that if we could get these particular special atoms into my crystal, we would be able to solve the structure because the physics said I would. You know, so I think that's where it did help.
We thank Venki for another time for a fantastic lecture. And remember, the Ribosome exhibit on the 4th floor is open. The museum closes at 9:15, so if you'd like to go take a look at that, do that quickly and enjoy the rest of your evening. Thank you.